Hello, I'm Alex Satz, and I'm going to talk to you about the new WuXi HitS platform. So first, let me talk a little bit about what we believe it means when we say target to hit platform. So first, if you start on the left-hand portion of this slide, it means getting the protein made. And of course, the correct protein needs to be made in order to conduct the screen. And after a screen is done, to find new hits which interact with your target, those hits need to be confirmed to determine if they are indeed real in biophysics assays. Now, after those assays are done, you could take those hits and generate a hit package. And within that package would be a series of hits that interact with your target and perhaps binding constants for those hits. But of course, if you're going to give this hit package to a medchem team and they're going to optimize that in order to potentially create a drug, it would be good to be able to have a crystal structure, because with a crystal structure in hand, the med chemist can work much more quickly. And of course, if they can work more quickly, they can cost less too. So since all of these different parts are connected, and of course you can't do the screen without the protein or the physics follow-up or the crystal structure, we certainly at WuXi thought it would make sense to bring all this expertise underneath a single roof and to create a single team which can do all of these things. Now, of course, when you do a lot of different things, it means that the size of your team needs to be quite large. And we have almost 200 FTEs working for us as part of the HitS team. On this first slide here, I'm showing you the management part of this team. And of course, you can always go back through these slides and look at them a bit more. I have one more slide showing not really the management group, but the scientific management group. And just as a proxy for introducing everyone else, I will just show you who I am. If you look on the bottom left of this slide, that's me, Alex Satz. All of us who work on the HitS team, we have a lot of know-how in this field. So for instance, I've been working in the pharma world for almost 20 years. I've worked as both a med chemist and a screener for numerous pharma. And I've either worked on the hit to lead part or the screening part of, I would say, close to 100 different drug discovery targets. So all of us, if you went through these slides, bring a lot of wealth of know-how in our specialty. So we have 4 different sites around the world. If you start on the left-hand side of this slide, we have the Krelux site. They specialize in making proteins that are very tough to make. They do fragment screening, forming the crystal structures, and biophysics follow-up work. Suzhou can make proteins as well, and Shanghai is probably our largest site with 130 FTEs, and most of the DEL work is done there. So the DELs are both made and screened. And then we have a very small but growing Boston site, which currently has 4 FTEs. So oftentimes what might occur is that a project team comes to us and they have the idea of a target that they would like to screen. Now, they don't actually have the protein made. And so if a project team comes to us with just a target in mind, we can do all the follow-up work in order to create a hit package at the very end. So that would include making the proteins, the screen, the follow-up work, and the crystal structure. Of course, every project team has different needs. Some project teams, the protein will be in hand and they'll only want the screening to be done. And you could imagine they might want different things. So when the project teams come to us, we can work with them to decide what they would like to do for us. So of course, we could do the entire workflow, or we could just do bits and pieces depending upon what you would like. So with the rest of these slides, I'm going to attempt to convince you that we have expertise in all the different things that we claim to be doing. So we'll start off with the proteins. Our Krelux site has had many, many years of expertise in making a lot of different targets. We have produced over 1,200 of them, including over 250, which are crystallography grade. The team as a whole enjoys working on targets which are very tough. So if they're If your team is having a hard time getting a protein made of the quality that needs to be made in order to do the follow-up screens, then you should try us out. Okay, and then, of course, after the protein is made comes the screening step. We specialize in binding-based screens. So certainly WuXi as a whole does do HTS, traditional HTS screens. We specialize in fragment-based screening and in Dowels screening. So if you've never heard of Dowels, I will come back to that topic later. At the moment, let me talk a little bit about the fragment screens. We can conduct these fragment screens by MST, SPR, X-ray crystallography, or NMR. So we have many different options depending upon what the project team wants, and perhaps even more importantly, what the protein itself wants. To talk about that a bit more, the library itself that we screen has over 3,000 members in it. We have made this library collection just for us. It's designed so that whatever hits we find, those hits are going to be things that a med chemist would actually want to work on. They are rule of three compliant, and they're designed to cover as much of that rule of three space as possible. And of course, things that are considered to be, a pain to work with, and others do not exist in our screening deck. Speaking about the fragment-based screen, this is the workflow for MST and for SPR. What's the most important point of this slide is that if the protein is in hand, we can conduct the screen and give you the results from that screen in about 4 weeks. So the turnaround time is quite short. We can also conduct the fragment-based screen with using a soaking system so that every hit which is found, you will have a crystal structure for. Or we could conduct the MST or SPR screen ahead of time, call out those hits which we find are real binders, and then we can use that and the soaking system to find crystal structures for all of those hits. Okay, as I said, I would talk about Dell a little bit more. What I'm showing you here is a generic picture in order to help understand what a Dell screen is. So on the left-hand side of this slide is shown the barcode. And the barcode encodes the chemical structure, which is shown on the left-hand side of this slide. And the chemical structure and the barcode are connected by this flexible linking chain. So the reason why Dowels are often used now is because you can make these Dowels, you can make a lot of different structures rapidly and at a relatively low cost. And this is done because we can use split and pool combi chem. So I want to give you an example of how this is actually done. So take, for instance, the blue box here, which has 3 different things in it. You could read off any one of the barcodes, and by knowing the sequence of the barcode, you would know what the chemical structure was on the left-hand side. So if you take that mixture of 3 things and you split it up into 3 tubes, that's a split size of 3. So it's split and pool combi chem, and we have just now done the split part. In each one of these new tubes, you add 1 BB, which does chemistry on the left-hand side, and you add 1 tag, which you ligate on, making a longer barcode on the terminus of the barcode. You now create 9 new products. Each one of those products is encoded by its barcode. You can take those 9 different things and you can combine them into a pool. And it's okay that you have a mixture, because at any point in time you can sequence the barcodes of what exists in that pool, and you know what is there. So on the previous slide, I talked about using a split size of 3. That split size is very small. We like to use and build our Dowels using split sizes of 96 or even split sizes of 9,000. And in that way, a single FTE over the span of a month can make millions and millions of different chemical structures. And of course, we can then screen those structures against your target to find out which ones bind to your target. At WuXi, we have over 40 chemists who only make Dowels. And so we have made over 200 Dowels. And we can take those Dowels, all these different Dowels, and combine them all into a single tube as shown here, so that we can actually send you a kit. And in each one of these little tubes, you have 14 billion different structures. You can actually, in this case, run that screen in your own lab, so that even if you wanted to do a Dell screen and you didn't want us to know what the target was, we could send you this kit and you could do the screen. Of course, we also have the option where you send the target to us and we do the screen. So how is this screen done? It is a binding screen. The target is immobilized on a resin. And then if you go to the second panel, you then take the Dell. So the Dell is simply the liquid from one of these tubes. You add it, and library members which bind to your target stay, but library members which don't bind to your target can be washed away. If you go to the third one, you can now elute the ligands that were bound to your target by running a heated buffer over them. The heat causes the protein to denature, and now your protein binding pocket is gone. So you collect those things which were bound to your target, you sequence the barcode, and that tells you the structure of the ligand that was bound to your target. And in the end, as many pharma companies have found out, they can run these Dell screens and find hits, which can be progressed into the clinic or into different kinds of in vivo models. And I give a couple of citations here of things that you can look into further if you. The key to finding good hits is to have good Dells. And we make our Dells with over 35,000 BBs, and we also have over 6,000 specialty ones designed just for us in order for use in making Dells. And these are BBs which you cannot purchase from any outside vendor. And with Dells made in this manner, we have a much better chance of finding hits that med chemists would want to work on. So with our team, we have made over 200 Dells. We can make over 10 Dells per month. And of course, if your project team wanted to make a special kind of Dell for the target, which they wanted to go after, we could, of course, do that as well. We can make a Dell in about 4 to 8 weeks, so that process is very fast. And as I said, we can screen targets which are sent to us, or we can send a kit to you, and the targets can be screened by you and your lab. We also offer an option which is shown here on the right-hand side for academic groups, and this is a free way of screening if you're an academic group. So after you do any kind of a screen, you need to do the follow-up work to make sure that the hit is indeed real. We have a very long list of ways that we can do this. I feel like the simple take-home message from this slide is that if you have a target and you have hits, we will find a way to determine if those hits are indeed real and if they are indeed binding to your target. Okay, and lastly, it's very hard to do medchem unless you have a crystal structure. You can't always get one, but if you can, you certainly want it. We have a lot of expertise in how to do this. We have established conditions for over 150 different targets. And so we have a turnaround time in that case of about 8 weeks. If it's a demanding target and a protocol is published somewhere else, then we can get a crystal structure in about 4 months. Or we also can do work in cases where no known structure is out there. We do about 10 cases of these per year. And of course, that's always fun to work on the really tough stuff. Okay, so I'll leave you at last with this slide, which outlines all the different things that we can do. I've already gone through all of these point by point. And I will just say thank you for tuning in, for downloading this, and for listening. Thank you very much. Bye.